I have no disclosures, sadly. And want to thank the San Jose Surgical as well as the Department of Surgery here at Stanford. And you've already heard about the contributions of Dr. Armanini, and appreciate the support and the invitation to come and speak in his honor, and reflects how important it is that what we provide, what we do, often lives on long after we're gone. And that's an important lesson. And we all have influence on our trainees that lasts for lifetimes. And so it's critically important that to honor those who have come before us. And hopefully I have also contributed to that. I thought a lot about what to talk about as coming back here, and this really has felt like a homecoming. I know so many of you, which I've known for decades, and kept in touch with all of you. It's so great to see so many people here who have had such an influence on my career. Appreciate Dr. McDougall coming, from internal medicine and radiation oncology, and has taught me so much when I was a junior faculty member here about taking care of thyroid diseases. And so many more of you who have had such an influence on my career, and I appreciate all of you making the effort to come here today. So I thought I would talk about the life as a surgeon scientist and reflect a little bit on what I've done in the past. And although to some we might seem like mad scientists, as depicted in the picture here, but I thought I would start out by giving a little bit of an update on how I chose the areas that I focused on in my research, give you a little bit of an update from what I've done since I left Stanford with the discoveries that we've made. And then I was going to talk a little bit about the challenges facing surgeon scientists, as well as talk a little bit about future surgeon scientists and some of the successes we've had in the recent past on training the next generation of surgeon scientists. So there'll be a little bit of, for everybody, the first part of the talk is for those of you who speak gel, that'll be there, but then we'll go on to other things too. So bear with me if I'm not clear on what I'm talking about. So when I finished when I was in medical school and finished medical school, this is an example of the kind of things that were being published. Here's a graph showing patients with breast cancer, and those that expressed estrogen receptor had a much better overall survival than those that didn't. And I found this to be extremely intriguing, and wondered why would this be? Why does expression of this estrogen receptor change the clinical phenotype of these tumors? And so one of the very first things that I did when I started here at Stanford as a faculty member was to say, well, let's look at the promoter for the human estrogen receptor. And so we cloned the promoter of estrogen receptor and made the standard, 5' deletions, and then deleted these parts of the leader, and then cloned them express them into ER positive and ER negative cells, ER positive T47D and ER negative MDA and B231, and then assayed them. And as you can see here, not much happened as we deleted the whole region until we went all the way down to the start site. But then, interestingly, this little region here that was in the untranslated leader, we noticed that when you deleted this part from 210 to 130 plus 135, all of a sudden you lost expression tremendously in ER positive, but no real change in ER negative cells. So we went ahead and took that small region of DNA, we cloned it, radiolabeled it with P32, mixed it with cell extracts, and lo and behold, got a factor on a gel shift, which was present in the ER positive cells, but not ER negative cells, which we initially called ERF1. So this was interesting, thought maybe this might be controlling expression of estrogen receptor in breast cancer, and could be a key to understand why those patients do better than patients that don't express it. And so we went on over the next many years. We cloned this from MCF7 cells. I say we, it's the royal we. It was actually Lisa McPherson, who's the first author on this paper, who was able to show that purified from about 3,000 plates of MCF7 cells this factor originally called ERF1, and then was able to show that it was actually a novel AP2 transcription factor in the AP2 family. So as shown here, we proved that, and it's been confirmed by many other people, that if you knock out TFAP2C, ER will go away. And the related factor, AP2 alpha, encoded by the AP2 alpha gene, knock it out, it doesn't really change estrogen receptor. So it was very specific to this transcription factor, which is, and I apologize, but it's called AP2 gamma. It's encoded by the TFAP2C gene. So when I use both those interchangeably, I'm referring to the same exact thing. And we went on to show that it is also involved in driving oncogenesis of luminal epithelial cells. If you knock it out in ER-positive cells, the cells actually go through EMT, and they turn on genes like CD44 and vimentin, and I'll show you just a tiny bit of that data, and as well as its ability to, its expression and the genes that it regulates have significance in terms of predicting outcome in other kinds of breast cancer, such as HER2-positive breast cancer. We then more recently wondered about the role of AP2 gamma in mammary development, since it was involved in oncogenesis in luminal epithelial cells. So we used a mouse model with a conditional knockout of the TFAP2C gene, which is the mouse homologue of the human gene, and used an ER fused to Cre, which is inducible by tamoxifen, driven by the keratin 5 promoter, as shown here. So here's the floxed, exon 6 of the floxed AP2 gamma in the AP2 gamma gene. And you can see here when you treat the cells with tamoxifen, it drives expression of Cre into the nucleus of the cell. And so we could pulse these animals for about a week just during puberty to knock out expression of this gene. And we use a dose of tamoxifen that we and others have shown has no effect on the development of the mammary gland. But when you knock out AP2 gamma expression, we had a highly significant reduction in ductal outgrowth from these cells. And so one of the things that we concluded this was doing was also controlling mammary gland development. So to try to understand how it was doing this and what it was doing, we used single-cell RNA-seq from animals that were treated with either a vehicle or tamoxifen, pulsed them with tamoxifen. And one of the things that we were able to find is that we had a significant reduction in this cluster 4 within this mammary gland. So you do this clustering based on the patterns of expression, and that defines different subtypes of cells within the mammary gland. So loss of TFAP2C gene induced a reduction in this cluster 4 cells. We used something called pseudotime analysis to try to understand what cluster 4 was. One of the things, the pattern of expression suggested it might be mammary stem cells, and in fact, using this slingshot program to analyze what's called pseudotime, and we can get into this, it's kind of a crazy concept, but you can begin to tell what these cells may have come from. And this cluster 4, we found, gave rise to both basal cell clusters as well as luminal cell clusters. And so we believed it was a keratin 5 positive mammary stem cell. And so the model that we were able to derive from these data indicated that this embryonic mammary stem cell gives rise to a keratin 5 positive AP2 gamma positive cell, which then gives rise to these other luminal and basal cell clusters. So this whole mammary development is very closely related to oncogenesis, which makes sense, and we're finding that in a lot of other systems as well. And so this keratin 5 population, we believe, was dependent upon AP2 gamma expression for maintenance of its mammary stem cells. But we needed to prove that knockout of TFAP2C actually alters mammary cell function. And one of the ways that we did that was we took animals with the FLOX gene, isolated mammary epithelial cells from them, and then infected those cells with either an adenovirus expressing GFP or an adenovirus GFP Cre. And as you can see, when we knock out the TFAP2C gene, we no longer were able to form mammospheres. In addition to that, we were able to show that those mammospheres, or the cells when you knock out TFAP2C, also inhibited reconstitution, meaning we took cells or tissue from the animals that were treated and put them into a cleared fat pad of a syngeneic animal and then looked for mammary gland development. Here are tissue fragments, and if you recover them from tamoxifen-treated animals, they are unable to form these nice mammary gland structures. In the same thing, we use single-cell transplantation, where we took these cells basically that could form these mammospheres and inserted single cells into the mammary fat pad. And if the cells were treated with CRE, they didn't form that, whereas GFP, they formed these nice glandular structures. So the conclusion here is that this is involved in controlling mammary gland development and the luminal phenotype. But there's a little bit of a conundrum here. As I told you, this AP2 alpha, which is highly homologous, 83% similar, 76% of the amino acids are identical. And it's another transcription factor in the same family. It's also expressed, but doesn't appear, it seems to control a completely different set of genes. So how could that be? As shown here, if you knock out the TFAP2C gene, knock it down, all these luminal genes change and go down. But when we knock out TFAP2A, it doesn't happen. Knock out TFAP2C, these basal genes like CD44, MMP14, they all come on, but knocking out A didn't do that. So you could say, well, it's just a dead transcription factor, but that's not the case, because genes like P21, CDKN1A gene, when you knock out A, it goes away. Knock out C, it doesn't. So these highly homologous factors regulate completely different sets of genes. And so how could that be? Well, the first question was, what about where they actually bind? Do they go to different places in the gene, and AP2 alpha goes to the P21 gene, but AP2 gamma goes to the other? So we did something called ChIP-seq, which is just a technique to ask where in the genome do these transcription factors sit? Where they occupy? And as you can see here, AP2 alpha, we're using an AP2 alpha antibody or AP2 gamma antibody, have identical patterns of expression. And this was true all over the genome. So we said they're binding to the same place. And of course, the reviewers at Cell basically said, well, your antibodies cross-react. Well, we didn't think that, so what we did to then is we cloned both AP2 alpha and AP2 gamma with HA tags, expressed each one individually, and then used anti-HA. And what's shown here is where each of these red dots are. These were where we looked for amplification. And in fact, whether we put in C-tagged or A-tagged proteins, they bound exactly the same, confirming that they went to exactly the same place. So how could that be that two transcription factors are bound to the same location, but one regulates this set of genes, the other regulates a different set of genes? So to try to figure that out, again, we wondered about how we could figure this out. What we ended up doing was we made chimeric proteins. They're so homologous that it allowed us to take, here's AP2A, here's AP2C, and what we could, since we lined them up, we just took parts of each one and created sort of Frankenstein, pieces of that and the other piece of the other one, and then assayed each of these in cells looking for what was regulating luminal genes. And long story short, if it contains this head of C, even if the rest of the molecule is TFAP2 alpha, it acts like C. But you can take all of AP2 gamma and just put in the little bit of the head of the activation domain from AP2 gamma, and it will look like gamma. Wherever the activation domain comes from is based, it will control what genes it actually will regulate. So we said, okay, how does that work? So we used a technique called yeast-2 hybrid to begin to find what proteins were interacting with AP2 alpha and AP2 gamma. So defined a set of genes. Then we tested them through this rather complex assay of knocking out each one individually and asking which one of these factors, in fact, when we knock them out, will upregulate genes in the luminal category. So, to make a long story short, again, there were only 2 genes from this entire screen that acted the way we thought it would help us understand how AP2 alpha and gamma have this differential activity on luminal gene expression. As it turns out, these 2 genes in the screen are PIAS1 and UBC9. Both of them are 2 genes in the SUMO pathway, which didn't seem like it could possibly be random. So we believe that SUMOlation must be controlling activity of AP2 alpha. And in fact, AP2 alpha is SUMO conjugated. There's a lysine 10, which is SUMOlated, which is just a little peptide. SUMO peptide gets enzymatically stuck onto this transcription factor, and that completely changes how it functions. And if you mutate lysine 10 to an arginine, it can't be SUMOlated, and that makes it SUMO resistant. And what we're able to show is that if we took these cells, these are MCF7 cells, basically these are MCF7 cells, they make estrogen receptor, they don't make CD44, and they make these enzymes in the SUMO pathway. If we take MCF7 cells and we create a stable knockout of TFAP2 gamma, ER goes away, as I just showed you, CD44 comes on, so this is what I've already showed you, but interestingly enough, if we knock out these enzymes in the pseudo pathway, all of a sudden ER comes back on and CD44 goes away. So SUMO inhibition, we believed, allowed AP2 alpha to rescue the loss of AP2 gamma with regard to regulation of luminal genes. In addition, if you put in the K10R mutant, which can't be SUMOlated, back into those knockout cells, it will now be able to turn on luminal gene expression, whereas the endogenous kind of gene, if you just put in a wild type AP2 alpha, it isn't able to do that. So clearly SUMOlation is altering how this transcription factor functions. As I've said, one of the things it's done is it inhibits CD44 expression, and this is a variety of cell lines shown here. In addition, we used a patient-derived xenograft, which is a sad story. A woman came to see me in clinic. She had a fairly significant palpable breast mass, and she said it wasn't there 3 weeks ago, so I just did a core biopsy in clinic, and it came back as a basal breast cancer, in which she got chemotherapy and just continued to progress on chemo, so I did a mastectomy on her just to get local control. And she eventually died, and what we were, with her permission, got the cells from her tumor and created a xenograft in a mouse. And these are actually her cells here, where they are almost completely composed of cells that are CD44 positive, CD24 negative, which defines a stem cell phenotype in breast cancer. And if we treat these cells, either these established cell lines or her cells, with ginkgolic acid or anacardic acid that are SUMO inhibitors, you can almost completely eliminate this stem cell population. And we were able to show that this whole effect was dependent upon AP2 alpha. Just not to go on for this, but in any case, if you treat these cells with ginkgolic acid or anacardic acid, you can repress CD44 expression, but if you simultaneously knock out AP2 alpha, then that effect goes away. So this regulation of what we believed was stem cells was also dependent upon this transcription factor AP2 alpha. And so what we were able to show is that we inhibit the ability for these tumor cells to form a xenograft in nude mice. That's a stem cell or tumor initiating cell kind of assay. Interestingly though, we found that after about 3 or 4 months, animals that had been treated with anacardic acid do get tumors. Interestingly, we were able to find that those tumor cells that come from an animal that was treated with anacardic acid almost completely lacked these cancer stem cells. And this assay was done using aldehyde and aldofluor and CD44. This is the stem cell population here. Goes from 83% to 0.3 with anacardic acid. So we hypothesized that these tumor cells would not be able to form a secondary tumor, which is an assay for stem cells, in naive animals. And in fact, that turned out to be true, that there are heritable changes once these cells are exposed to anacardic acid. And these cells derived from an animal that originally saw anacardic acid never formed tumors even after a year, whereas readily the tumors derived from animals that were just treated with vehicles will form secondary xenografts. So that's an assay for a tumor initiating or cancer stem cell. And what was interesting is we found similar effects in basal breast cancers as well as in colon cancer cells. More recently, a resident working in our lab was studying colon cancer cells with knockout of this gene, where we used CRISPR to create a complete knockout of this, and identified a number of target genes in colon cancer that this transcription factor is regulating, and suggested that it was regulating cell cycle, which turned out to be true. When you knock out AP2 alpha, you increase S phase, it prolongs S phase, and it induces a resistance to PI3 kinase inhibitors. And one of the things we're doing now is to use the expression of AP2 alpha as a marker for response to PI3K inhibitors. Interestingly, in a melanoma model, we recently have been able to find that AP2 alpha also controls melanoma metastasis. Again, one of the things we did was use CRISPR to create this panel of melanoma cells in A375 melanoma that failed to express any of this AP2 alpha protein. Interestingly, when you put these into a SCID mouse, they form primary tumors but failed to metastasize. So although there was no effect on the growth of the cells in culture and no effect on their ability to form primary tumors in the animal, it completely lost the ability to form metastasis. And so we luciferize these cells, and you can see that here. These animals get, with the A375 parental cells, they get liver and lymph node metastases, but with AP2 alpha knockout, these animals fail to get metastases. So, and again, identified a similar prolongation of S phase that seems to be related to this metastatic effect. And by trying to understand how this is working, what we were able to do was use something called BioID, where you basically fuse biotin ligase to your bait. The bait here was the AP2 alpha protein. And then you ask, okay, what is being biotinylated? And that means that that protein must be in close proximity to the protein that you attach the biotin ligase to. And what we happen to notice is that many of the factors that were pulled out in this screen were members of what's called the NERD repression complex. And we were able to confirm that by co-IP, as well as showing changes in target gene deacetylation for the proteins that appear to be regulated by AP2 alpha. So the model that we created was this, shown here. This is the NERD complex. AP2 alpha is somehow closely proximated to this location, appears to be controlling it through the DNA binding domain of AP2 alpha, and it blocks activity of the NERD complex. If you knock out AP2 alpha, it's gone, and the NERD complex now deacetylates these histones and turns off a variety of genes. So we concluded that AP2 alpha regulates members in the E2F family through inhibition of the NERD repression complex. One of the genes that it regulates is called EZH2, and we had a number of experiments to demonstrate that EZH2 regulates the effect of AP2 alpha in its ability to form metastases. There's a recent drug that was developed, which is a specific EZH2 inhibitor called tazemetostat. And what we were able to show is that drug tazemetostat reproduces the same effect as the ability to knock out the AP2 alpha gene. What's shown here is, in fact, these animals, it looks like there's more all this fluorescence that's up above this level of fluorescence is all due to metastases. But if you treat the animals with tazemetostat, you completely eliminate these formation of these metastatic nodules within the liver and within lymph nodes. And this reproduces what AP2 alpha knockout was doing in those cells. And it's also heritable, meaning that if you recover tumor cells from the primary tumors of animals that were treated with tazemetostat and put them into create secondary tumors in naive animals, they no longer are able to form metastases, whereas the animals that were just treated with vehicle clearly continue to have metastatic ability. We also used a mouse model of melanogenesis. These are mice that you can induce melanomas in, and we use single-cell RNA-seq cluster analysis to analyze both melanocyte stem cells in these animals as well as the melanomas that are deriving from those melanocyte stem cells. And what we were able to find by single-cell clustering was that there is this interesting cluster here which is developed from these melanocyte stem cells that you see in the melanoma and have high degree expression of TFAP2A or AP2 alpha based on that cluster analysis. And this very same cluster also overexpresses EZH2 as well as other family members in the E2F pathway. And so one of the models we're working out is the fact that AP2 alpha controls metastases, and it appears to replicate what is normally happening where these where melanocytes actually move to their location, and that's dependent upon, if you eliminate AP2 alpha, there's a paper that came out a few years ago showing that you can eliminate the ability for melanocyte stem cells to migrate to where they normally are in the skin. So this is the model of metastasis. This is my last science slide. So this is the model of metastasis that we put together, is shown here. It's AP2 alpha. Inhibits the NERD complex, EZH2 is turned on and induces metastasis. You can knock out AP2 alpha. It then represses EZH2, so metastases go away, and you can recapitulate that by treating with a drug that blocks EZH2 directly and eliminate metastases. So I'm going to move on to the second part of my talk, which is to talk a little bit about surgeon scientists and make the case that they are critically important for these translational kinds of studies that I just went over with you. I think that there's a critical role for surgeon scientists to help advance our understanding, particularly of things like solid organ tumors, which is really a disease process that is largely managed by surgeons. I mean, we're primary care physicians for breast cancer as an example. And here's an editorial that was published in Nature 4 years ago that talked about a critical need for surgeon scientists, dangerous cuts, number of surgeons involved in research is falling. And they find that this is a problem. Increasingly, surgeons are turning their back on research. And their conclusion was that policymakers must stop seeing surgeons as easy sources of clinical revenue and really support them to also be scientists. As you all, I'm sure, are aware, our investment nationally in research has continued to decrease since the 1960s, and our non-defense R&D spending of the federal budget is less than 2%. Now that's in contrast to other countries, as you know, most notably, for example, South Korea here and China shown here, where there's been a tremendous increase in investment in R&D, and the U.S. is not increasing their investment at the same by all the same rate. And it really has been 2 decades of stagnant NIH funding. This is dollars in terms of investments in NIH, and this is in using inflation-adjusted dollars. You can see there's been a tremendous decrease trend over the last 2 decades for NIH funding, as well as the fact that the amount of funding per grant has lagged over the last 2 decades. And that results in an extremely low success rate for NIH grants. And a success rate for NIH grants is down around 20%. For NCI, it's even lower than that, single digit, somewhere around 8%. So this really has created a difficulty with continuing our research efforts in the United States. And another dangerous trend, which is shown here, is that shows the number of percent of NIH research grants with principal investigators who are under age 35, which is tremendously decreasing, and an increasing percentage of those over age 66. I guess for those of us like myself, they'll be 66 in another year or so. You know, maybe that means that my grants will have a greater success chance in another year. But again, I think this is a very dangerous trend and maybe having a serious effect on the pipeline of investigators. And funding to surgery departments is particularly troublesome. What's shown here is NIH funding in total dollars, not considering inflation, showing a statistical increase over the last 10 or 15 years. And funding to internal medicine departments is at least maybe a trend in increasing, but certainly isn't decreasing. And this is compared to funding for surgery departments, which has continued to demonstrate a statistically significant decrease over the last 15 years. And this was also highlighted in a number of articles. Here's one from the Annals of Surgery from 4 years ago showing that surgery department NIH funding relative to total funding or to internal medicine is a statistically significant decrease in funding to surgery departments. And this is associated with and likely causative of a decrease in basic science abstracts submitted here. It's the, talking about the Academic Surgical Congress is where these data come from. And so I think this is a dangerous trend that needs to be called attention to. Because of this, there has been increase in industry-sponsored research in the United States. And as you see here from the 1950s and 60s to 2017, a tremendous increase in funding from industry, whereas federal funding continues to lag in this. So a lot of surgeons have also turned to industry-supported research. That could include big pharma, biotech as a source of funding, as well as many surgeons funded through medical device companies to support their research interests. So there are a number of motivations for this academic-industry relationships. The motivations for people on the academic side is, well, it's a source of funding for the projects that you're interested in. It gives you an opportunity to test practical application. You might be able to gain unique insight into the systems that you're working with. There may eventually be business opportunities that come from these relationships that we have with industry. On the industry side, they're motivated because it gives them an opportunity to work with brilliant individuals in universities like Stanford, and they can solve technical or design problems, helps them develop new products. It gives them access to physicians who see patients and potentially can then investigate clinical application of their discoveries, allows them to reorient R&D, as well as gives them access to recruitment. And there's a number of individuals who get recruited out of academia to work in industry. But there are challenges with working with industry. The first is there can be a loss of compromise of your independence. And so if you begin to work with a company, you have to understand what strings are attached. Are you going to be able to publish your research findings? If your research findings show that the product that the industry is developing doesn't work, are you going to be free to publish that? So you have to understand what all the relationship means that you have with industry. There can also be inappropriate commercial exploitation that goes on that could tarnish the reputation of the organizations and institutions that we hold dear. It also can create conflict of interest between patients and economic self-interest of the physicians. This cartoon says, try this, I just bought 100 shares. And that can also lead to erosion of the public trust. And I'm sure all of you have seen in the news, you know, even dealing with this COVID pandemic, there's a lot of distrust of physicians even in terms of recommendations to get vaccinated. And some of this comes from this kind of erosion of public trust which has happened with physicians who were always in the past regarded as those that you should believe and take their advice seriously. And it underscores the need for an IRB and other oversight. So what are the others, besides industry, what are the other sources of funding? Well, there are a number of societies that offer research grants for surgeons and surgeon scientists, as well as some non-surgical societies. As you heard, Stanford just got one of these, the American Surgical Association Foundation Fellowship. It's $75,000 per year for 2 years. It funds young investigators without previous NIH support. And I disclose I was a recipient of one of these when I was on the Stanford faculty. So Stanford has done very well with having their faculty supported through this kind of foundation grant. Other than the ones that will be announced this week, hopefully, there were 69 previous fellows awarded, and those individuals who have had this support from the American Surgical have a strong track record of funding. American College of Surgeons has a $40,000 2-year fellowship. The American College, along with the Society of Vascular Surgery, gives support for a KO8 supplement. There's the George H.A. Clues Award also that I had when I was here at Stanford, which was very helpful to help jumpstart my research career. There's the National Surgeon Scientist Program, where half comes from the college and half from the department of the individual. And I want to draw your attention to the ACS Board of Regents Innovative Grant to expand diversity, equity, and inclusion and anti-racism. They give out a number of $25,000 and $50,000 grants to both individuals and institutions to support DEI and support of underrepresented minorities in medicine. Society of Universal Surgeons has a mid-career award, which is a $40,000 bridge funding award. There are junior faculty awards for underrepresented minorities in medicine, as well as the Research Scholars Award from the Karl Storrs, which supports digital technology in the OR and surgical innovation. There may be opportunities through a variety of foundation grants, which are shown here, and many other smaller foundations that you may be able to use to support your research. So I'm going to share with you my own personal formula for success, which is kind of tongue-in-cheek, but it ends up being true. 999 failures, and I've had them all, plus 1 success equals success. So you have to keep trying. Said another way, from Thomas Edison, I have not failed, I have just found 10,000 ways that won't work. And so the key here is you have to be persistent. You can't give up. And surgeons are individuals who face the challenges, and I believe that if you are persistent with time, you will be successful. In addition, the formula to success is, in my opinion, having a home support structure and that your partner in life is as committed as you are to what you have done in terms of committing yourself to academic surgery and particularly being a surgeon scientist. Paula, unfortunately, was not able to make this trip, as she's shown here, but almost always comes with me, and particularly if I ask. Here she is with the American Surgical, and when I was president of the SSO. I met her here at Stanford, so I had a lot of things happen to me at Stanford that were life-changing, this being probably the most significant, other than our children being born here. And we got married on June 19th. And those surgeon scientists in the audience know what that means. I was going on a honeymoon for the next 10, 14 days. I wasn't on call. I didn't have any cases in the OR. I had no clinic patients. And so it allowed me to work on my grant renewal that was due on the first week of July. And Paula accepted that, which is, so important that your partner in life appreciates what you do and is supportive of that. And you have to build a team. Here's a group of the folks that worked in my lab, hosted here at our house, a dinner party, and it's so important to have outstanding individuals who can actually do what you're planning on and have a great committed team of individuals. One of the things that we've had that have helped develop the future of surgeon scientists is our T32 in surgical oncology research. Shown here are a number of one of the first couple classes of individuals that were supported on this. Phil Sponheimer did his fellowship in surgical oncology at Memorial Sloan Kettering. He is now at UNC Chapel Hill. Jenny Carr also trained and then stayed with us in plastic surgery and is now on the faculty at UNC Chapel Hill. Jennifer Robbie did her colorectal fellowship at Cleveland Clinic, and now we were fortunate to recruit her back. Jess Maxwell did her surgical oncology fellowship at MD Anderson, is now on the faculty at MD Anderson. James DeAndrade did his surgical oncology at City of Hope, and we recruited him back. And Scott Sherman at the University of Chicago did his fellowship in Surg-Onc, and we also were able to recruit him back. And some of these individuals have worked in the lab. I showed you some of their data in the presentation that I presented. We have a number of individuals who graduated from our residency who spent time in the lab funded by the T32 and are now in fellowship. Vincent Wu did the work on HER2 that I pointed out. He is now is a breast fellow at Columbia. Matt Alexander is doing pediatric surgery at Columbia. Anthony Swatek is with us doing cardiothoracic surgery fellowship. Allison Lorenzen, plastic surgery at UT Southwestern. Kendall Keck, plastic surgery with us. And Justin Wilkes, surgical oncology at Moffitt. And this 3 individuals out of our 6 chief residents spent 2 years in the laboratory. Anna Beck is doing her fellowship in breast fellowship at Memorial Sloan Kettering. Really outstanding. She's going to be the next Mike Longacre. I think she's got like 40 publications already as a chief resident. Aaron Scott is doing a surgical oncology at City Hope, and Ed Cho is going to stay with us for another year and then probably going to do minimally invasive surgery. And a number of our senior residents who also spent time in the lab, Dakota and Kelsey were in my lab, and then they did some of the results that I showed you of the complex analysis of doing single-cell RNA analysis in a number of our systems that we're working with. And then finally, this is my last slide, our current Iowa research residents are shown here. I do want to point out that Femi Shiraju was not able to be funded on our T32 because he's a Nigerian citizen, but he was the recipient of the SUS Resident Research Award this year. I'm very proud of what he's already been able to accomplish in the lab, and want to draw attention to that. He received the award out of 52-something applicants and was the individual there. So for our T32, I think we're contributing to the pipeline of future surgeon scientists. 29 residents completed a 2-year research fellowship. 38% of these individuals were women, 10% were Latino, 17% were Asian, 7% Black or African American. And so my final comment is that we are making progress. And I think that we need to continue to invest in developing surgeon scientists. And I think it's critical for our future in surgery that we do so. So I will stop there and thank you all for your attention.